Welcome back to the Deep Dive, where we synthesize the most important knowledge from a stack of sources, articles, studies, and research to give you a genuine shortcut to being well-informed.
Today, we're embarking on a mission into, well, the fundamental biology of existence.
Yeah, how life maintains balance.
Exactly. We're diving deep into the intricate machinery that keeps your internal world stable, whether you're, facing a tiger or just dealing with a Monday morning.
And our sources today are pretty fascinating. They range from foundational anatomy, the endocrine system, all the way to the latest in mathematical modeling.
Right. Models used to predict how your body handles chronic stress.
So our focus today is really biological communication and regulation. We're going to cover the endocrine system, the famous stress response, the HPA axis, and the fundamental rules, things like feedback loops.
And the goal really is to move past just simple definitions and show you how these complex integrated systems all work together.
What's so interesting to me here is seeing how this complexity is now being treated almost like an engineering problem.
It is. We're not just exploring what hormones are, but why these integrated systems, like the neuroendocrine axis, are so delicate and, frankly, so challenging to stabilize.
And that challenge, crucially, impacts our health. It links to everything from anxiety to metabolic disease.
It really does.
Okay, let's unpack this with the basics first. The endocrine system. I like to think of this as the body's broadcast network.
That's a perfect analogy. Unlike the nervous system, which is like a private, lightning-fast text message to one recipient.
Right.
The endocrine system is a chemical radio broadcast. It's made up of all the glands and tissues that produce hormones, are chemical messengers.
And they're secreted directly into the bloodstream.
Directly. And they travel throughout the entire body, but they only act on specific target cells that have, the right receptor antenna to pick up the signal.
And that contrast in speed is so critical to understand. The nervous system is, what, milliseconds?
Oh, incredibly fast, using both electrical and chemical signals for very localized, very rapid effects.
Whereas the endocrine system, it's slow.
Much slower. It relies only on chemical signals, so it takes seconds, sometimes minutes, to respond. And the effects can last for days.
But, and this is the key takeaway for you, despite those huge differences, they both serve the same ultimate purpose.
Homeostasis, maintaining that internal equilibrium.
Okay, so let's get into the chemistry, because a hormone structure basically determines its entire career.
It really does. How it's made, how it travels, where it acts. We've got three main classes. First up, the peptides.
These are the amino acid chains?
Yep, chains of amino acids, which makes them water-soluble. Classic examples are things like insulin, ACTH, TSH.
And they start out as these much bigger molecules, right?
Exactly. They have this whole manufacturing process. They get cut and refined in multiple steps before the final hormone is actually released.
And here's where a little detail becomes super useful, clinically speaking.
Yes, because that synthesis process means the final hormone is often co-secreted with its leftover bits, its cleavage products.
Like with insulin and C-peptide.
Right. They're released in equal amounts. And this is a crucial nugget for clinicians because they can measure C-peptide levels to get a precise, reliable marker of how much insulin a patient's own body is producing.
So it's like a window into the factory floor.
That's a great way to put it. It's essential for diagnosing and managing diabetes.
Okay, moving on. Next up, we have the steroid hormones. These are the fat-soluble ones.
Right, derived from cholesterol. They're lipophilic, fat-loving, and since they're lipid-soluble, they can just cruise right through the oily cell membranes of their target cells.
So things like cortisol, aldosterone, estrogen.
Big players, yeah.
And finally, we have the tyrosine derivatives, which kind of play both sides.
They do. They're all derived from the amino acid tyrosine, but they're function splits. You have the water-soluble ones, the catecholamines.
Epinephrine and norepinephrine, the fight-or-flight stuff.
Exactly, from the adrenal medulla, the instant response molecules.
But the other key derivative, thyroid hormone T3 and T4, that's different.
It's the exception. Despite being from amino acid, it's highly lipophilic, more like a steroid. It needs special carrier proteins to travel through the blood.
And its synthesis is pretty complex. It needs iodine, which is why iodine deficiency is such a global issue.
A huge issue.
So let's move from the chemistry to the coordination. Hormones rarely work alone. How do they interact?
The interactions are layered. You can have an antagonistic effect where one hormone does the opposite of another.
Like insulin and glucagon with blood sugar.
Perfect example. One lowers it, one raises it.
Yeah.
Or you can have a synergistic effect where two hormones together achieve something greater than the sum of their parts.
Then there's the permissive effect. This one is fascinating. One hormone doesn't do the main job, but it has to set the stage first.
It does. A good way to think about it is that the first hormone, the permissive one, is basically putting up more receptors on the target cell.
Ah, so it's making the cell more sensitive.
Exactly. Is plugging in the power cord, so when the second hormone arrives with the actual message, the signal is way stronger.
So the cell's sensitivity isn't fixed. It can turn the volume up or down.
Absolutely. If hormone levels are low, cells might use upregulation, adding more receptors to catch every little signal.
And if the levels are too high?
Then you get downregulation. The cell starts pulling receptors off its surface. This is a huge mechanism behind things like insulin resistance in type 2 diabetes.
So how does the message actually get inside the cell?
Well, again, it depends on the class. The water-soluble ones, peptides, catecholamines, they can't cross the membrane, so they bind to receptors on the cell surface.
Often G protein-coupled receptors?
Yes, and these act like intermediaries. The binding activates internal second messengers like CAMP-MP.
And this is where the signal gets amplified, right?
Massively amplified.
Yeah.
That one hormone binding outside doesn't just trigger one internal event. It sets off a cascade that can amplify the signal thousands of times over.
Which is why a tiny amount of a hormone like ACTH can have such a huge effect on the whole body.
Precisely.
Meanwhile, the lipophilic hormones, the steroids and thyroid hormone, they just ignore all that.
They do. They just diffuse right across the cell membrane, find their intracellular receptors, and the whole complex goes straight to the DNA.
It becomes a transcription factor.
Right. It binds to specific DNA sequences and literally changes which genes are being turned on or off. It alters the cell's long-term behavior.
This all brings us back to control. How does the body know when to stop?
That is the negative feedback loop. It's the master control mechanism of homeostasis. The manufacturing analogy is perfect here.
Right, if the stockroom shelves are full,
The product itself sends a signal back to the factory floor to slow down production.
it maintains equilibrium by shutting itself off.
Whereas the positive feedback loop is the opposite. It's rare and inherently unstable. The product just increases its own production rate.
Like oxytocin during labor.
The classic example. Contractions lead to more oxytocin, which leads to stronger contractions, until the whole system terminates with childbirth. It's built for speed and a clear endpoint.
Okay, let's focus now on the ultimate integration of all these systems. The stress response, governed by the HPA axis.
The hypothalamus pituitary adrenal axis. This is the central highway for managing stress. It starts in the hypothalamus, which is command central.
And it releases CRH.
It releases corticotropin-releasing hormone, CRH, and importantly, arginine vasopressin, AVP, which is a co-stimulant. Those travel down to the pituitary gland.
Which acts as the dispatcher.
Yep. The pituitary then releases adrenocorticotropic hormone, ACTH, into the circulation
And ACTH travels to the adrenal cortex.
and tells it to make and release cortisol, our main stress hormone. Cortisol then feeds back to the hypothalamus and pituitary to shut them down, completing the loop.
And this axis isn't just waiting for a stressful event. It has its own powerful rhythms.
That's what's so brilliant about it. First, you have the circadian rhythm synced by the master clock in the brain, the SCN.
So cortisol levels change throughout the day.
Dramatically. They're lowest late at night, but they start rising toward morning, peaking between 6 and 10 a.m. That spike is the famous cortisol awakening response.
It's physically preparing you for the day.
For the day's metabolic demands, exactly. And then layered on top of that is the ultradian rhythm.
Okay, what's that?
The HPA axis doesn't release hormones in a continuous stream. It releases them in pulses, about 2 to 3 times an hour.
So when you're stressed.
The frequency of the pulses might not change, but the amplitude, the height of each pulse, increases dramatically.
For a long time, our view of stress was really shaped by Cannon and Salai.
Right. Cannon gave us fight or flight, this idea of a single unified adrenaline system. And then Hans Selye popularized the general adaptation syndrome.
Alarm, resistance, exhaustion.
And his big idea was that the response was nonspecific. Didn't matter what the stressor was, the body's reaction was broadly the same.
But that view has been fundamentally revised.
It has. Which brings us to this idea of primitive specificity.
So what does the modern view tell us that Salai missed?
It tells us that the stress response is not a single gear. It's a tailored strategy. Different stressors create measurably different patterns of response.
Can you give an example?
Sure, if you're exposed to cold, you might get a massive norepinephrine response. If your blood sugar is low, you get a huge epinephrine and ACTH cortisol response.
So the body isn't just hitting the panic button, it's deploying a specific ratio of messengers for that specific threat.
It's a highly coordinated specific deployment. And this distinction is so crucial.
Why?
Well, because it changes how we think about treatment. If every stressor produced the same chemical signature, we'd have one drug. But if the pattern is specific,
Then maybe specific psychological traumas leave a specific chemical fingerprint.
exactly, a specific balance of CRH, AVP, cortisol, which could lead to highly targeted therapies down the road.
This specificity also brings us to a concept that really refined homeostasis, allostasis.
Yes. So homeostasis is like a thermostat set to 70 degrees. It always wants to return to that fixed point. Allostasis means achieving stability through change.
It's more like a dynamic navigation system.
Perfect way to put it. It sees the environment it's changing and proactively adjusts the optimal set point to meet an anticipated demand.
Like your blood pressure rising before a big presentation.
Right. Your body elevates its baseline in anticipation of that challenge. That new elevated state is the allostatic state. It's adaptive, but only in the short term.
Because the body isn't meant to live in that state. That's where the cost comes in.
That is the allostatic load. It's the cumulative wear and tear. If your internal navigation system is constantly anticipating an emergency and keeping the dials running hot,
The systems wear out.
they suffer decreased efficiency. And chronic allostatic load is strongly linked to hypertension, metabolic syndrome, inflammation. It's the physiological price of continuously adapting.
Speaking of that wear and tear, let's talk about the nuances within the HPA axis itself, especially the role of the hippocampus.
Right, the hippocampus, which is central to memory, plays a key role in modulating that CRH release from the hypothalamus.
So cortisol has to bind back to the brain to complete the feedback loop.
Yes, but it uses two different types of receptors there. The mineralocorticoid receptors, MR, and the glucocorticoid receptors, GR. And they don't do the same job. Well, the MR receptors seem to be saturated at lower baseline cortisol levels, and they mostly contribute to the strong negative feedback that slows the system down.
Okay.
But the GR receptors, they're only fully engaged during high stress. And in some brain regions, they sometimes appear to contribute to positive feedback.
Wait, so they can drive the system harder?
Potentially, yeah. When acute demand is highest, it's this high wire balancing act between two different receptors that makes the whole system so difficult to stabilize in chronic conditions.
Let's bring this back to the clinic. What happens when this precise balance fails?
The clinical picture is dramatic. With cortisol, think of Cushing's syndrome, which is from way too much cortisol. It leads to immune suppression, metabolic chaos.
And the opposite is Addison's disease.
Right, hyposecretion. Not enough adrenal steroids. It causes catastrophic electrolyte imbalances and a total inability to manage stress.
And of course, the pancreas gives us our big metabolic disorders.
Diabetes is the benchmark. Type I is just an absolute lack of insulin. Type 2, a failure of balance caused by that downregulation we mentioned. Insulin resistance.
Plenty of insulin, but the cells just won't listen.
They refuse to listen to the message.
Now, we also need to briefly touch on the other big communication systems, lymphatic and immune.
This is a huge field now.
Cool.
Neuroimmunoendocrine crosstalk. The immune system is a major endocrine disruptor.
How so?
Well, inflammation, the body's second line of defense, signals the hypothalamus. Immune messengers, called cytokines, can directly stimulate CRH release, driving that HPA axis.
So stress and immunity are intimately linked.
Incredibly so. Under stress, your body often diverts resources to the adrenal system at the cost of immune function.
That puts chronic stress in a whole new light. But is this kind of chemical language unique to animals?
Surprisingly, no. It's universal. Plants use phytohormones, their own endocrine system, to control everything from how they grow to how they handle a drought.
Okay, let's look at the growth promoters first.
You have auxins, which make the main shoot grow tall, and cytokinins, which promote cell division and are famous for the Richmond-Lang effect,
Which is?
delaying aging or senescence in plant tissues, keeping them green longer.
And the gibberellins.
They cause that extreme stem elongation, making a plant bolt. They're also used commercially to make seedless fruit, like in tomatoes.
So those are the go signals. What about the stop signals, the ones for managing stress?
On the inhibitory side, the big one is abscisic acid, ABA. It's legitimately nicknamed the plant stress hormone.
Why is that?
When a plant senses drought, ABA is released, and it causes the stomata, the little pores on the leaves, to close, conserving every last drop of water.
So it promotes survival over growth.
Exactly. And then there's the odd one out, ethylene.
The gaseous hormone.
Right, it's a gas that promotes ripening and senescence. It's literally why one bad apple in the barrel ruins the whole bunch. The ethylene gas it releases triggers ripening and decay in its neighbors.
Wow. From the neuroendocrine loops managing our morning cortisol spike, all the way down to a plant hormone closing a leaf pore. It's a vast chemical landscape we've covered.
And it's all controlled by this precise biological signaling, all designed to maintain some kind of equilibrium.
Looking back at the models you mentioned, it seems our ability to predict HPA axis dynamics is getting better.
It is. It's improving significantly by incorporating those nuances we talked about. The hippocampal feedback, the MR versus GR receptors, the role of AVP.
That move towards specificity is encouraging.
It is. But the mathematical models still have enormous gaps. They struggle to incorporate things like individual genetic and epigenetic differences. And maybe most importantly,
An emotional component.
the powerful immediate role of the amygdala in processing and transmitting emotional stress signals. We're still missing the emotion in our chemical equations.
So, what does this all mean for you, the listener, navigating a world that feels so stressful? Since we know chronic stress forces the body into an altered steady state, leading to allostatic load, that accumulating wear and tear how many of our daily physiological experiences, from unexplained fatigue and disrupted sleep, to persistent anxiety, to even common metabolic issues, could fundamentally be traced back to our body's continuous attempt to regulate around a perpetually altered, stressful baseline.
That's the real cost of imbalance.